All right, welcome everyone. Hope you enjoyed the unanticipated break. First thing I want to do is toast, although it's coffee, not champagne, to Sachs, to Leonard and everyone on the 10th anniversary. I think Mike and I have been coming almost since the beginning, so that's fun. 10 doesn't include COVID years, right? So this is subtracting out. All right, good. So, well, the first panel set us up really, really well. I'm just going to make a few introductory comments and then let each of our panelists introduce, and then we'll get into it. I suspect some of you may be a little befuddled by the title and what it actually means. And what it actually means is supposed to be something that's twofold, right? Actual new endpoints, and also including biomarkers in there, but also a concept that was brought up very specifically by Ben from Stifel when he asked the question on the prior BD panel, which was how to think about balancing risk, right? Technical risk, commercial risk. This is one of our bugaboos at Lumanity, and we've posted a lot about it. So, but you heard a lot of themes set up that will, I think, resonate through this panel in that prior session in terms of, for example, Neil's comment about, we don't need another CD19, CD20, TROP2, et cetera, or Devang's comment about how it's unlikely that, all 4 or 5 plus oncology agents in the same class, will all be successful. Of course, the footnote to that would be success is going to be defined differently according to different companies, as someone also said on that panel. But what we're going to be talking about, again, is really the two concept of kind of the conceptual aspect of thinking about endpoints and meaning and how to approach them, and then the actual literal idea of what might be some new endpoints to pursue and new markers. So with that, you know who I am. So let me go to my left and Colette.
Hi, everyone. My name is Colette Lipp. I'm part of the business development oncology team at Merck KGaA Darmstadt, Germany, known as EMD Sorono over here in the US, part of Gregor's team for those who were present in the previous panel. I've been at Merck for a number of years now, spent quite a bit of time in the business development team, both in the technologies and in the oncology space, was responsible for Merck's transactions in the targeted protein degradation space with emphasis to Proxygen and recently C4 Therapeutics. And also was responsible for the repatriation of Bavencio from Pfizer last year, as well as the Hungary deal that Gregor also mentioned on the PARP1 inhibitor and the Claudin 18.2 ADC, and also had the privilege of working together with a few colleagues on the Abisco transaction on pimacotinib, the CSF1R inhibitor. I don't think I need to go into too much detail on what Merck is interested in. I think Gregor covered this nicely this morning, but we're, of course, interested in ADCs, in oncogenic signaling, DDR, and immuno-oncology, preclinical all the way through to commercial. So looking forward to the panel today.
I like the use of the word repatriation. It's good. All right, Jim.
Good morning, Jim Barlow. I'm the CEO of ImmunoGenesis. My background is in clinical and commercial strategy, focused in the oncology space. Was at Merck for 8 years and BMS for 3, and then for the last 10 have been in biotech. With Inovio Pharmaceuticals and Genios Therapeutics. At ImmunoGenesis, we are developing therapies that can unlock the potential in immuno-oncology. And specifically, we have a PD-L1, PD-L2 inhibitor that's engineered with effector function to clear out immunosuppressive elements, because we strongly believe that there's a lot of room for improvement in the PD-1 space and that you need a better monotherapy that works in things like colorectal and ovarian. So I'm definitely representing the proven pathway, improved approach space for the last discussion.
Matthew.
Good morning. I'm Matthew Price, co-founder and chief operating officer of Promontory Therapeutics in New York, and sit at the intersection of clinical development, R&D, CMC, and regulatory, and also external relationships with collaborators, among whom have been Pfizer and EMD, Merck Darmstadt, and also currently the National Cancer Institute. And our approach is within small molecules, looking for and developing ways to engage the immune system and the anti-cancer immune response through intracellular anti-cancer activity rather than T-cell engagement. So this leads to a number of interesting outcomes. I can refer to the public data sets generated by the National Cancer Institute, where we see immune responses using liquid biomarkers and solid tissue approaches, and an absence of immune-related adverse events. in thymus cancer patients. That is obviously a rare disease where we're partnered with NCI. In our lead phase 2 program, we're working in a late line patient setting of metastatic castration-resistant prostate cancer, which is effectively a post-standard of care setting, and have fully enrolled our phase 2 in the United States and in France with 111 patients treated, with submission at ESMO and hopefully also at ASCO GU early next year.
Great, thank you. Nadim?
Good morning, Nadim Ahmed, CEO of Cullinan Therapeutics, which up until about 6 weeks ago was Cullinan Oncology. So we've made a strategic expansion into immunology through a CD19 by CD3 T-cell engager, which we're very excited about. I've worked at GSK, Celgene, BMS through the acquisition. So I've sat on both sides of the deal table, so understand what the strategics want. And then, we're biotech can partner up. So I think that's a little bit of a unique view. As a company, we take what we call a modality agnostic targeted research approach. So our discovery efforts always start with the target first. So we identify the high impact targets, and then we decide the best way to prosecute those targets. So for us, it's always target first, modality second, which is different to some of our peers, but it does mean that we're not relying on a single platform or even a single product. So if you look at our pipeline, quite a diverse range of indications across oncology, immunology, as well as modalities. Excited to be here.
Great, thank you, Nadim. And perhaps, as you pointed out, Raven, the youngest company on this panel.
Thank you. This is Raven Lim. I'm the CEO and the co-founder of the company Pilatus Biosciences. The company is actually a spin-off from the Ludwig Institute of Cancer Research in Lausanne, and also the scientific studies backed by the Cancer Research Institution in New York. So our focus is through the immune metabolism. This is it's not really new, but it's a new approach which we do believe can target a potential current unmet need in the immuno-oncology space. So my background is in playing the civil head of business development and corporate development, and I have a 2 IPO experience before, particularly in the IO space. We also team up with several oncology companies like AbbVie and several big pharmas. Okay, looking forward to our conversation. Thank you.
Thank you, Raven. All right, so let's start with the most practical aspect of new endpoints. So, I'm curious to hear from the panel how each of you may be using or looking at companies that are using or deploying. That's particularly for you. Novel endpoints or biomarkers. So, for example, MRD, well established in some settings, but not in others, say myeloma versus AML. Clearly, complete pathologic response, validated clinical path, high risk advanced breast cancer and TNBC, but not necessarily elsewhere. And of course, there are things like ctDNA and CTs themselves, et cetera, that people have been looking at. And the reason why these may be interesting, aside from what's brought up in the prior panel, was potentially issues of stratification, but also perhaps acceleration of development, which of course for a biotech in particular can be very critical. So I'm just curious to hear again from each of you kind of how you are or are thinking about or might be using any of those or others. Again, some of those are concepts, some of those are biomarkers and specific targets. But I'm going to go now this direction, from starting with Raven.
Okay, so our lead molecule is targeting to the CD36, which is played several important roles in all the metabolic disease and also the metabolic environments. But our approach is to use this CD36 can in one way, disarm the Treg and also boost the T cell. So we found that's a very interesting target is that this target can also play a very important role in all the immuno-oncology space, and that this CD36 can be a good biomarker. Based on the current results, we found the high expression of CD36 also correlated to some prognosis cancer disease, such as ACC, such as CRC. And we have several ongoing development plans, particularly in silicon analysis, to really understand why CD36 can play this important role, and also to address those the current tumor inhibitors, to find out some of the synergy. So this is still ongoing development for the CD36 endpoint biomarker. Yeah.
And that's, of course, a very common going after overexpression of a given target, whether HER2. But of course, there are many other situations where target expression level is not really relevant. You don't necessarily even need to do the analysis because 80-90% of the patients express. So I'm curious, again, particularly Nadim, with your comment about being target centric and modality agnostic, how that is playing into your thinking.
Sure. I mean, one of the markers we're looking at, but in very different ways, is MRD, or minimal residual disease. And so in the AML setting, it does create a window of opportunity for you to explore very earlier line settings. So for example, the frontline setting post-consolidation, which typically we start with end-stage patients and work our way up. MRD and AML allows you to get into that frontline setting quite quickly and get the results quickly. So we're very excited about that. We have a T-cell engager, CLN-049, which targets both FLT3 and CD3, which are both, FLT3 now is clinically and commercially validated. So we're excited about that program. And we're also interested in the application of MRD, which is newer in multiple myeloma. So, we had the recent AdCom where there was a unanimous vote. When do you have unanimous votes in AdComs? But that was exciting to see. And so that could be a new endpoint for both late stage and early disease in multiple myeloma. So we have a monoclonal antibody, CLN-619, which targets a novel pathway, the MICA/MICB pathway. It's the first program in the clinic. We saw single agent efficacy at ASCO last year, and we're presenting data at ASCO this year in the combination approach. So MRD we're very excited about, but we continue to look at things like ctDNA, et cetera, as well.
Yeah, I may want to follow up if I remember, and we have time, on the MICA/B target. Sure. It's an interesting, with particularly broad applicability.
That's right.
Potential. So, Matthew?
So our approach is largely focused around pharmacodynamic monitoring. So as an example of that, within prostate cancer, there really is, at least at the mCRPC setting, there's no one assessment that you can do that can capture the disease effects and the treatment effects in the patient. So our approach has been to cast a wide umbrella of different tools in order to see the evidence within biomarkers of clinical effects, of immune effects, and of reductions in tumor shedding, or CTC, circulating tumor cells. We do have sampling ongoing for ctDNA analysis, which will be quite exciting to us, which I think is really moving towards the front in terms of the ability to find a more global measure of disease response in the blood in disease settings where conventional tools simply do not capture the totality of a therapeutic effect in a way that will really predict for ongoing or long-term clinical benefit. So prostate cancer's complexity, in other words, has caused us to deploy a number of tools that we think match the drug's mechanism and what it can do in this setting. So those are, as I think a question will come up, are any of those validated? Are any of those regulatory endpoints? What we did do is make one of them a secondary endpoint, namely CTCs. And the reason being that there is a wide body of literature, even meta-literature now, in about 6,000 patients with mCRPC, where analyzing reductions in circulating tumor cells has been shown to be equally or even more predictive of overall survival benefit than conventional markers like PSA or even RECIST reductions of tumor size. So when you read that and when you learn that, you say, we need to do this, we need to look at this. But in honesty, I think the field is moving more towards broader approaches like ctDNA, and so we're very excited to get those analyses done in the near future as well.
Jim?
Yeah, I think I think it's a real balancing act because there's more and more pressure to show efficacy data early to be able to drive funding, and at the same time, there's a lot of skepticism as well on that early data. So I think it's important to stick to some of the traditional endpoints because you need to leverage all of this historical data and historical controls that are out there. So I think that having the traditional endpoints front and center is key. And I mean, we saw with the Gritstone example where they tried to use a unique and for where they were trying to show proof of concept, but use a unique endpoint. And I think it ended up backfiring, which is, you know, it's, I give them credit for trying to do that approach, but it just shows sometimes the difficulty with that. Our approach is to try to look at early on some biomarkers that can inform future development or give us a hint of where the optimal patients might be. So there's a molecular amplification 9p24.1 that is a hallmark of Hodgkin's lymphoma, where you get overexpression of PD-L1, PD-L2, and JAK2. But it's actually quite prevalent in solid tumors as well, about 30% of triple negative breast, and quite a bit higher percentage than MSI high. So we're going to look at that endpoint because it's ideal for us. Our targets are expressed from tumor stem cells out through the blasts, and the tumor microenvironment tends to mirror that expression. So we think that could be an ideal patient group for us. So we're going to look at that because that could really inform perhaps a pan-tumor type approach in future trials. So I think it's that delicate balance of trying to get as much as you can out of that early stage data to drive funding and using those traditional endpoints, but then also trying to enhance with other approaches.
Great. All right. So Colette, you get a chance to kind of synthesize, summarize, and give your own perspective after those.
Yeah, I think from a kind of BD perspective, and I think I really agree with Jim here that ultimately, I think, you know, maybe it may not accelerate having these kind of, you know, novel endpoints, but of course, anything that can inform on the program and give confidence obviously adds, you know, value for potential partnering or investors and things like that. However, ultimately, especially from a partnering perspective, if, you know, the partner who you're going after then has to sort of, you know, repeat things because you have actually decided to, you know, not go after traditional endpoints, then ends up, I think, potentially backfiring on potential, you know, partnering opportunities there. So I think that from a pharma and from a partnering perspective, I don't see them as sort of, you know, surrogates. They can't be, you know, these traditional endpoints can't be replaced. But of course, if they can give confidence, you know, potentially drive the development path, give, you know, sort of more details around that, then I think that of course it adds value. But at the same time, I think pharma also really looks to biotechs to obviously pave the way here. I mean, ultimately, this is, you know, this is always going to be an evolving situation, and, you know, some novel endpoints today won't be novel in, you know, a few years' time. So
yeah, that's an interesting point though, because it puts a lot of pressure, time and money on the biotech, whereas pharma is the one that's better resourced and probably better, you know, staffed in terms of departments to kind of develop and move those new
but
endpoints.
potentially more risk averse.
Yeah, I maybe not even potentially. All right, so now let's open this up a little bit, because it's one thing to either focus on standard known endpoints or biomarkers versus developing new ones, but one way or another, you know, you still need to make a judgment about how the information that comes out from those, again, either standard or novel, influence your decision about what to do with that program. So given that, I'm going to jump straight to you, Nadim, to kind of talk about this issue of rigor, right? Because I think we talked about in this panel, and you know, at Lumanity, we've posted a lot about this, about the fact that, you know, when the biotech world was so flush with cash, it's not clear that the best decisions, trying to say this as diplomatically as possible, were being made because you didn't need to, right? There was the illusion of an infinite runway, which of course was an illusion. So Nadim, with a little of that setup, please, I'm curious to hear your comments.
Sure. Look, that's a very good point. So we have a pretty high bar for go, no-go decisions from early in the discovery process through to the clinic. And so we front load as much experimentation as possible in the least capital intensive phase, which is preclinical. And there we do that through what we call thriller or killer experiments. And so when we get the killer result, as the name might suggest, we kill the program quickly and we reallocate the resources. When we get the thriller result, we say, okay, how can we double down investment and how can we accelerate this program because it looks very promising? What we don't do is change the goalposts after we get the results. So my team, I'm very proud to say, is very disciplined about doing that. And so I can give you an example from my big pharma days, and many of you may have experienced this. I'd be at a governance committee meeting where we terminate a program, and then 2 years later, I find out some people are still working on that project. I'm like, wow, I thought we killed this program, right? But our team is ruthless like that. And I think that's really important because it does start to de-risk your efforts as you start to go into the clinic. And that's the advice I would give. Don't change the goalposts, have high bar, go, no-go decisions. And then that way you're taking both clinical and commercial risk into consideration as you move into the clinic, which is obviously the highly capital intensive phase of drug development. So that's something that we're very disciplined on.
Great. And so I'm going to now open it up for people to kind of comment on that. And I'll just throw in one kicker just to be more controversial, right? There's part of that rigor, particularly in the less capital intensive phase of preclinical, by definition, is dependent on model systems. And we know the endless debate about how relevant or not animal models are in oncology. It's actually kind of a silly because it's very dependent on what models, what situation, what target, et cetera. So the nuances are important. But please, everyone else kind of weigh in with their thoughts about this idea of rigor and especially that front loading concept.
I think what popped in my head there is one of the reasons I think biotech is sometimes more successful at developing therapies than big pharma is because we tend to be focused on one or two things, and when you do hit challenges, we work around them and improve them, and we keep going, and we don't kill things. Whereas I think big pharma may, if they hit a challenge, may kill it early. So it's kind of interesting. So I totally understand where you're coming from, Nadim, and maybe it's different because you said like you start with target, so you maybe come with different approaches versus if you start with an approach and you're looking at target. But I think that's kind of an interesting balance. Is I do think part of the nimbleness of biotechs is you have a program, you believe in it, you've shown that you believe it has some preclinical proof of concept, and then when you hit some bumps, you sort of work around that.
Yeah, I couldn't agree more. I mean, the last thing that we can do is validate a new endpoint as an individual, private biotech company. So we have to think differently. And I think to Jim's point, the thought process is, okay, we see efficacy. We have an indication where we've seen it. We believe in our mechanism. We have a drug, right? We don't want to kill it if we continue to see the right things. What tools can we deploy to make sure that we're seeing the things that we need to see in a way that can become convincing? So it really is, it's an interesting point. It's perhaps a different mindset. And so we've done a lot of work in literally building the relationships with the people who are developing the tools. So Howard Scher is a great example on the topic of circulating tumor cells. He's been very outspoken about this. He's, somebody who's led the charge with big pharma, with FDA. So we've partnered with him as one of our lead investigators, and he's now the head of biomarker development at Memorial in New York. So for us, it's about finding the pockets where the tools are well honed. And similarly, on the front of ctDNA, we're speaking with a very specialized lab now that has a focus and a very rich historical data set within prostate cancer specifically. And then on the immunoprofiling side, I forgot to mention earlier, who better to do this with than the National Cancer Institute? You cannot have 150, analytes in multiplexing as a standalone private biotech company. The National Cancer Institute has that. So I didn't realize this was going to be my answer, but the mindset of finding the expertise in the tools has been a big part of. Thanks for the prompt, Jim.
Well, I mean, it's an interesting. I mean, I think it is a kind of a dynamic balance between kind of this proposition of thriller or killer. Now I just have Michael Jackson dancing in my head, versus this idea of kind of, if you're a true believer, and of course there are good and bad aspects of that phrase in the molecule or the target, trying to figure out how best to showcase that without, again, like making it live forever when it's not going to do anything. Like, we worked years ago, including Mike, my colleague, on flavopiridol, which could never be killed at Aventis, is probably still poking around somewhere at Sanofi. But before I go to you, Raven, I'm just curious to hear from you, Colette, about this dynamism and tension between kind of these concepts.
I think ultimately that, the amount of investment that pharma companies make in, these programs, I think this thriller or killer approach doesn't work so well there, right? I mean, I think that ultimately everybody wants to find a solution if they can, obviously, to keep the program going and try and, resolve any kind of challenges that pop up and things like that. But ultimately, it's obviously a bigger engine at the end of the day, right? There are obviously higher, strategic decisions that need to be made, and resources that need to be diverted elsewhere and on different programs. And so ultimately it's more often than not going to be an investment and a sort of a money decision rather than a kind of, trying to keep beating at the potential program.
Yeah, I think I think, Jim, to your point, because we've taken this target-driven approach rather than platform-dependent approach, I think some companies, you find when they've invested so much in a platform, it's kind of hard to walk away from it. We made the conscious strategic decision that we weren't going to do that. And I'm not saying that's the right decision or the wrong decision. We just chose to do that. So for us, it works better. And look, I certainly agree with my colleagues. If it's, for example, CMC things you can overcome, or there's a little bump in the road, we're not saying we won't, try and solve those problems. But what we want to make sure we don't do is just keep pounding away at something. Hope is not a strategy. And so that's why we try to be a bit disciplined about it. And I think, look, one very clear example of that would be, for us, monotherapy activity is really important. And so, when you see monotherapy preclinically, that doesn't necessarily mean you're going to see monotherapy clinically. But if you don't see monotherapy preclinically, you probably aren't going to see monotherapy clinically, right? So that's one of our kind of key go, no-go criteria. And if we don't see that sort of activity, what we don't want to do is invest, dollars and dollars and dollars behind a combination approach that's going to take you years to show the contribution of your specific molecule. So I do think there are, as Jim said, there are areas of gray, and it also depends on strategically how you approach it as well.
Yeah, and I think that issue of monotherapy activity came up in the prior panel. Of course, I've done many panels with Emmett Schmidt. That is the mantra, at least for Merck US. So Raven, to kind of come back around to you, young company, novel target, immunometabolism space that's, had some challenges in the past. Can you talk about kind of this idea of the rigor in the experiments and this issue of kind of single agent activity?
Yeah, so thanks, Nadim, set up the stage. So actually, we do believe that monotherapy is mostly important part. And because right now, we also set up very high bar in the preclinical mouse models. The first, we target into the immune responsive, the hot tumor part. If you see the monotherapy can also become tumor-free potentials, then we go ahead to push the cold tumor settings. And based on the data we generate, we also found a very interesting result is if the single agents can really treat the tumor, become some tumor reductions. And then we consider the several strategies, such as combined with the immune checkpoint and also the standard of care to really figure out the potential and how we address the current unmet need. For example, we have a very good data show that in the bring beta-catenin mutation in the SCC part, which is the really unmet need. And based on the results, we also found a very good encouraging data showing the 40% combined with the current standard of care, we can have a tumor-free. So those are kind of indicate that we also collaborate with very reputable hospitals who has a very good mouse model is the PD-L1 resistant models.
Yeah.
And based on these results, we targeting and try to position our molecules, the CD36 targeting immunotherapy after the standard of care failures. And combined with the reagents, maybe a PD-L1 or CTLA-4, we also have to set several animal disease model, which is very also important for us to really identify the potential need in the clinical. So we have a several approach to really identify the PD markers, to really understand what's the mechanism of action in line with our current findings in the immune profile changes. So those type of things we try to build the kind of evidence and decide have a go for no-go decision.
All right. So before we segue to
So can I comment on
oh
the
yes, please.
monotherapy?
Yeah. Of course, yeah.
No, I don't. I just the monotherapy thing is a real hot button for us because we believe that's the single number one factor of why immuno-oncology has struggled. Is so Ahmed from Merck, they've done these studies and they basically show that there is no synergy. That drug A and drug B, if drug A is 10 and drug B is 10, then you're going to get 20. You're not going to get 30. And so when he talks about that, I want to jump out of him because he says, don't even come to us to partner at Merck unless your drug has monotherapy activity. And then I want to say, yeah, but Emmett, isn't the problem that Keytruda doesn't have monotherapy activity in 55% of tumors? And that's why we believe you need a better underlying PD-1 that works by itself. And so this idea it just struck me that being able to show that early on is sort of a new endpoint in and of itself, right? Instead of these thousands of combination trials we've seen where you don't know the contribution of components, being able to show early on that you work as a monotherapy seems like a really critical success factor.
And of course, many of those studies, I can't put a percentage because I don't have it in front of me because it's a lot of data, are in what we'll call overlapping and redundant pathways, which is, I think, the more interesting point that Emmett makes from that analysis is that having non-redundant, or even some might call it orthogonal kind of pathways or approaches is far more likely to demonstrate that combinatorial value, whether true synergy or additive. But Colette, I'm curious to hear your thoughts on this discussion.
Yeah, I mean, I think that obviously, ideally, of course, we would want to see monotherapy activity, right? I think that, that would be always the nice to have. I mean, almost a must have, I would say also, right? Especially from a partnering perspective, if, someone is asking us to sort of put, the dollars down now, I think this is something that, is something that management always want to see. They always want to say, okay, is there, something behind this kind of combination hypothesis? And is there something, that we can really see already? And so I think that, and I think Gregor actually also mentioned this in the earlier panel, I think, we do a lot of combination studies where we provide drug supply. And I think this here for us, the bar is significantly lower. This is not so much of an issue if a biotech wants to go ahead and try and demonstrate this. But I really think for us to sort of put money on the table for a partner, this is, a really critical factor.
Great. All right. Any other comments?
I would just say it's important to define what that means specifically for a given mechanism and for a given disease setting, because there can be very monofocal concepts of what monotherapy activity means, namely a RECIST response rate, or in prostate cancer, PSA reduction, where these things mean different things in different diseases. I'll just give a couple specific examples. After a certain point in prostate cancer disease progression, PSA is only representing the residual AR-driven component of the disease, whereas there are multiple other drivers and multiple sites of disease, particularly the bone, where you need to be looking at different tools than just PSA or just RECIST. So it has to be clarified what we mean when we say monotherapy activity. And so for us, we've seen it, thankfully, but we also want to add these other tools to substantiate why we believe the mechanism is doing the things that we're hoping and, yes, claiming that it is doing. So it's a bit of an all of the above approach to, yes, you see a RECIST response, yes, you see a PSA response, but what else can substantiate the value? And so these other tools come to play as supportive evidence and as proof points of other aspects of what a drug can do.
And of course, we have seen and can point to even approved drugs where there is a disconnect between, say, RECIST-defined ORR and OS.
Yep.
So there are more things in heaven and earth than Ahmed has dreamed of, even. So I'm not picking on. I love doing panels with Ahmed. I'm invoking his spirit. All right, so let's shift a little bit now to this other concept about endpoints, which is, again, going back to what the question that Ben from Stifel made at the prior panel, which is how to think, particularly for biotechs, how to think about balancing that risk between technical risk and the potential trade-off that generally occurs with commercial risk, meaning those targets that are most well validated by definition have the most competition, and the bar is generally going to be higher for approval versus the trade-off of going after something that's less well validated, so it's more biologic or technical risk, but you're going into a potential white space. And, so I'm just curious here how each of you and your companies kind of think about that balance.
Sure. Yeah.
Nadim, we'll start with you.
Sure, happy to. So we kind of divide those two categories in terms of first-in-class opportunities and best-in-class opportunities. So I'll give you the example that I gave of AML. So we have a FLT3 by CD3 T-cell engager that we're currently exploring in relapse refractory AML. And there for us, FLT3 is both clinically and commercially validated. However, the difference is with the small molecule approaches, you're only targeting the mutated form of FLT3, whereas with a T-cell engager, you're actually using the extracellular domain of FLT3. So basically, you become agnostic to mutational status. So you can target both mutant and wild-type FLT3. So that opens up 80% plus of patients with AML for this therapy versus the 25% of, where you focus in on mutated form of FLT3. So there we believe we have a kind of a best-in-class opportunity. With MICA, MICB, we're first in class. So of course, for strategic partners, there's risk, right? It's untested, it's unproven, but we showed single agent efficacy last year in the phase 1 dose escalation study. So we're pretty excited about that program. Then we have something in between. I alluded to us changing the name of our company. So, I would say we have a best-in-class potential, first-in-class approach with our CD19 by CD3 T-cell engager. We've all seen the exciting data that came out from Germany with CAR-T CD19-directed approaches in lupus, for example, and other autoimmune diseases. As someone who spent a long time developing and commercializing CAR-T in my previous companies, they've been transformational in oncology, especially hematology. The application in autoimmune diseases may be very difficult, risk-benefit profile. So we believe that an off-the-shelf approach there with something like a CD19 by CD3 T-cell engager could open up many, many more patients. And so de-risk and also commercial opportunity there as well, because now you're taking the treatment to the patient rather than the patient having to go to a CAR-T certified center to get the treatment. And that was one example where there was huge external interest, where we actually chose not to partner despite some attractive term sheets because we felt we could drive value better ourselves, at least to the next stage of data generation. So I think for us, we kind of take first in class as the risk but reward opportunity, but best in class really focusing in on differentiation versus what's there before us. If it isn't differentiated, then we're not going to pursue it.
Well, and also, of course, you just mentioned and kind of a overlaying concept of a different type of balancing of risk, like taking it forward more yourself.
Yep.
Taking on a certain risk, therefore, but for a better value inflection at the end of the day than partnering it earlier than you might have otherwise. Others, comments? This kind of trade-off of technical and commercial risk.
I probably can add some comments. So basically, if you want to do the first in class, you always challenge. And if we talk about the endpoints, particularly the clinical endpoint, I think based on the current regulatory authority, they typically require this kind of OS or those type of things. But if you really think this is the opportunity that you want to address the unmet need, and then you should work with the kind of leading global experts. Perhaps, for example, we collaborate with MD Anderson's. They have a NASH-induced HCC model, which is not public yet, but this not published yet. But this model is to really, reflect the current disease progressions from the NASH fibrosis and to HCC. So we need to figure out if we want to target this type of really unmet need, is also the high risk, right? But in another way, for example, in HCC section, we also know that the first line is Viva plus the atezolizumab. But after the failure of this or resistant model incurs, the patient survive very well, but suddenly the disease progress, and then we need to figure out what is that the treating point we want to target. So we discussed with several global leading PI to really identify, can we play some role in that particular space? And if we success, perhaps we can communicate with the FDA to have a potential first track to really identify the current unmet need opportunities. So this is what we are doing at this stage. Yeah.
Jeff, I think also with risk, it can be contextual. So for example, aiming for POC versus registration can be two very different categories. And so one thing I always challenge my team is to think about the question, when is good enough good enough? Mm-hmm. Like, as scientists, as commercial people, we always want perfection, but you sometimes don't need it. So certainly for, registration-directed approaches, you have to have, pristine studies and make sure, you do everything correctly. Do you need 100% source document verification for an exploratory POC study? Probably not. So I think sometimes risk can be a little bit context dependent as well.
That's an interesting point you raised because, we have spent a lot of time with early biotechs kind of thinking through this issue of POC versus what the founder of several heritage companies back before Lumanity, Ed Saltzman coined, which is POR, proof of relevance, right? Which is, it's in a provable setting with a reasonable ROI at the end of the day. Because when you disconnect those two, then that means you're spending a certain amount of money and time just to demonstrate it works, but it may be a dead end, or you're not collecting all that would necessary if it actually were sufficiently interesting then to just continue into a registration path. So again, that's the idea of kind of that trade-off that comes on again, that biotech pretty much is forced to make when cash is constrained. Different mistakes when, as we said before, it's not constrained, but it is now. Other comments?
Yeah, I was going to say we're leaning into the the reasonably likely phrase of FDA. Everything that we're doing on the technical side is not a validated endpoint, as I described these PD monitoring tools. They, however, have been deployed along the way with an eye towards validating the program for the commercial unmet need, the commercial medical unmet need. And actually, it's been feedback from our discussions with large pharma that have really helped us, I think, do that. Because you can hear in those early partnering discussions where what kinds of questions are coming up. They relate very much to the last panel, and they actually do inform and help biotech in decision making, I think. So we've been lucky to have those early discussions, and it's helped us to choose the tools to try to further validate the proof of mechanism, the proof of concept, and eventually allow the conclusion that there should be proof of relevance in the program.
And I'm actually glad you brought that up. I was going to ask earlier about kind of what each of you has learned because again, there are many other nascent biotechs in the audience, from the feedback from not just prospective partners, but additional investors that it might be going after, in terms of particularly this issue of thinking about trading off of time and risk and money, right? Jim, any kind of gems to share with the audience from your experience?
Well, I was thinking about the first in class, best in class. I'm sort of laughing at if you want VC funding, maybe you want to be first in class, because as one VC said to me, we invest in novel, unproven technologies. The PD pathways 10 years ago.
Okay.
But I think if you look at the products out there, I mean, almost all successful products are really building on previous science. I mean, you look at HER2. I mean, there's so many examples, right, of where we thought we had satisfied area, and then you make that next leap in technology. And I mean, the immuno-oncology field is littered with these novel, unproven pathways that fell by the wayside. So, I'm obviously biased. We're taking a very proven pathway and trying to significantly improve on it. But I think that science evolves and it takes time, and there's been a lot of lessons over the last 10 to 15 years. And so learning those, taking those lessons, for example, Pfizer with and Merck with the Valuemab, I think hoped to have some killing function in patients, but didn't see that. So we ended up using mutations in our FC to try to drive that killing. And that's not a criticism of Pfizer or Merck. It's more just to say you learn through the science and seeing how these things evolve and take that next step.
I think we also have to be a little bit careful that we don't define outcomes or achievable outcomes as what you achieve with treatments today. And I think sometimes we get forced into that. And the unfortunate fact is that today, most patients with advanced solid tumors, despite all the progress, will die of their disease. And so checkpoint inhibitors, for example, have been transformational. Cell therapy has been transformational, but there are still many, many patients with cancer that are dying of their disease. And so I think you have to reframe, how you look at things and not be limited by what current treatments are achieving in terms of outcomes.
Absolutely. We spent a lot of time with our early biotechs thinking about what the future TPP might look like, not today's TPP. So, all right, we have only a couple minutes left. Questions from the audience? Comments?
So I'm interested, in your discussion of making a better PD-1, that you can drive more response, deeper response. And people have talked about this with CTLA-4 as well, right? That, you make a super effector positive anti-CTLA-4 and you're gonna drive better responses. I'm just wondering, what's the upper limit? Because we know that we know a lot about why people don't respond to a PD-1, and it's not, as far as I can tell, it's not often because you haven't given them enough drug. It's often because there's expression of integrins that activate TGF-beta, or there's an overwhelming LPA, LPAR immunosuppression, or VCAM. So how are you going to find those patients in which a little bit more anti-PD-1 activity is going to move the needle? It's a difficult thought experiment for me, and I would just love to hear your thoughts.
No, I appreciate it. It's great opportunity to talk about what we. So we believe in. There's two parts of it, right? One is in the philosophy of drug development. It's combinations are hard because of overlapping toxicities, and so if you can in a single molecule address multiple facets of the immune suppression, that can be key. And so our philosophy is in the immune excluded tumors, which is the largest subtype, that you have these barriers driven by MDSCs, CAFs, TAMs that keep the T cells out. And so the current drug, you can block all the PD-1 you want. If the T cells can't get where they need to go, they're not going to have an effect. So with our killing function, we believe we can take down that immunosuppressive barrier, allow those T cells to traffic, and then we also can block the PD-1 signal. So it's this multitasking ability that we think can unlock that efficacy. And then if you can be a 25, 30, 35% response rate in colorectal or ovarian, that's a really strong position to build on. We think one of the fallacies that's happened is, again, you take a PD-1 and say, oh, well, I mean, they're amazing drugs, so let's just throw combinations on top. But that's really hard to take 5 and 5 and make 30, and it hasn't worked. And so we believe you need something that underlying by itself is 25 to 30, and we think having this multimodal mechanism can do that.
All right. I think you can have a debate offline
Yeah,
afterwards. All right. There's a question
Sorry,
over there.
Sorry.
Thank you. Claire Waring from Cumulus Oncology. It's a comment and a question. Just a comment, just listening to all the discussions that I guess whether it's a validated endpoint, whether it's an unvalidated endpoint, what we're all really trying to do is generate data that gives people reasons to believe. So investors, physicians, pharma companies. And with that in mind, I'm just interested to hear what the panel, their perspective on biomarker-driven early discovery and development, because I think, the evidence is there to show that phase 3 trials where there is a patient subgroup or a patient population lifted out with a biomarker, the success rate at phase 3 is higher compared to an unselected patient population. And I just wonder what the panel think about building that biomarker-driven drug discovery and right from early drug discovery all the way through.
Thank you for that question. It was one of the points we didn't actually get to. So I guess we have a lot, even though we're over, we're going to take a little bit of time just to quickly answer this, and then we'll scuttle off.
Sure. So I'll comment on it from a late stage commercialization guy, Claire. So one, if you're going to do that, you want to know early as you think about access and pricing, all of those things. I also think the way to look at it as well is if you have a biomarker driven approach, ultimately, what is that opportunity? So, if it's, I'm going to use a silly example. If it's 3 patients in the world, that probably doesn't help you move the needle, right? But if you can find a patient population where you can see high response rates relative to a diluted response rate, that in of itself could be very, very attractive. And we do build in biomarkers to all of our phase 1 studies to help our go decisions. And then the final thing I'd say about that, kind of tangentially related, but a plea to all of my biotech colleagues is don't let our mission be dismantled by the IRA, which is a terrible piece of legislation, right? Biologics cannot answer the questions of science out there, everyone. And this is coming from somebody who has a biologics heavy portfolio. There's a room for small molecules, and we're seeing amazing advances. So please don't let that, dismantle. It'll get fixed somehow. But if you're working on a small molecule today, it has promise, don't let the IRA throw you off your stride. But coming back to your biomarker question, I think as long as you can define it early, it's clear, then certainly, to your point, you can get, much higher and better outcomes, but you do need to discover it early.
Colette, do you have any comments to add?
Nothing to add, to be honest. I think that ultimately, provided that, of course, if there's an unmet medical need and it, of course, brings, better clinical outcomes, of course, it makes sense, but ultimately it needs to be a commercially viable opportunity.
Yeah, that was going to be my only counter, is that pharma still would prefer not to segment the market. But all right, thank you, panelists. It was a wonderful discussion. Thank you for your attention. And Leonard, congratulations again on your 10th anniversary. All right.